So, gene sequencing is a very new technology, but it has come ridiculously far in a pretty short amount of time. So, the first ever organism to have its genome fully sequenced was the Haemophilus influenza bacteria, which is not the same as the influenza virus. And it had its genome fully sequenced in the 1980s. Then, the first animal to have its genome sequenced, the C. elegans worm, was in the 90s. Then, finally, in 2003, the first full human genome was ever sequenced. You may have heard of the Human Genome Project. It ran for years, and finally in 2003, we got the first ever human genome full sequence. So those were major technology breakthroughs. But now we have the NCBI database, which basically just has the full genomic sequences of millions of organisms. It's ridiculous. So all of these pretty much fully sequenced genomes of all these different things, between bacteria to plants to insects to mammals, all of that is stored in this online database, and you can find them using BLAST. So let's kind of go through that and see how this BLAST technology works, because I have to do it for a lab report and might as well make a TikTok out of it. So for my Wolbachia lab that I've mentioned multiple times on here so far, we isolated DNA from different insects, and then we sent those samples off to a much bigger, much more well-funded, actual professional lab to have the DNA sequenced. So this is what came back. We had 114 samples in it, but yeah, this one specifically is actually from my lab partners, so we'll use that one to do the BLAST technology. So what you can do, just Google BLAST, so BLAST, put that into the search bar, and it will come up with these results. After you click those results, you're going to want to go to Nucleotide BLAST. Click that, it'll bring you over to this. So kind of what we just want to do, we want to make sure it's in standard database, nucleotide collection, all that. Just make sure everything is set to the settings that we want. Then we want to copy paste this entire segment. So you want this stuff up here as well, all from that little arrow thing. We are going to copy that. Then we're just going to paste that into here, again making sure all of our settings are correct. You scroll down to the bottom, hit the BLAST button. It's going to load for a hot second here. Then what will come up is this page, and if we look, we see all of these scientific names come up, and then we can Google that and see what this thing is. Turns out that the insect that my lab partner had collected for the lab is Culicoides, one of the species, and it is a type of biting midge. So we know what this guy is, and then we can actually get a tree from it. Then that gives us this kind of phylogenetic tree. This right here, the highlighted one, is our sample. We can then download this bad boy into a PDF, then save this, and here we go is our phylogenetic tree, all ready to be put into my lab report. So essentially, as long as this species has already been described and put into the database, you can compare the DNA from your sample. You can just copy paste that whole sequence into the database, and if it's already in there, it'll come up and identify what organism you have. And then, if that organism is not in the database, you can put your own data in there, and that's the first instance of that organism ever been recorded in the database, and then that can be used by future researchers to compare if they find the same organism. So this is very cool, and it's just very interesting how far that this technology has come in 20, 30 years. The first one was done in the 80s, it's now 2023. That's a massive amount of progress. The first human genome was sequenced 20 years ago, and now we can just search up and find oh yeah, I got a fruit fly.